This is Chapter 5, Microbial Metabolism. So when we talk about metabolism, we're talking about all of the chemical reactions that take place in a living cell. So chemical reactions that help provide energy and chemical reactions that help create substances for the cell, things like proteins and other molecules to sustain life. So there are two types of metabolism. So your catabolic reactions are going to be your breakdown, and your anabolic pathways are going to be your building reactions. Catabolic and anabolic reactions are considered kind of like coupled reactions. So the products or the end products of one reaction is going to power our second set of reactions. So catabolism is a breakdown metabolism. So we're breaking down more complex molecules into simpler, more basic building block molecules. So these are going to provide the building blocks for our anabolic reactions. Catabolic reactions are considered exergonic, meaning they release energy. So it takes energy to form the chemical bonds holding this glucose molecule together. So if we break down this molecule into simpler components, we liberate some of that energy in the form of ATP. Anabolism is an endergonic reaction that's going to use energy to build more complex molecules from those building blocks. So think anabolic steroids that bodybuilders use to build muscle. So anabolism, anabolic reactions are building up new, more complex molecules. So they're going to need the energy that was produced by our catabolic reactions. So this is what I mean when I say they're kind of like a coupled reaction. So our first set of reactions provides the energy, and the ingredients are the building blocks for our second set of reactions. So showing another example of your anabolic and catabolic reactions. So with anabolic reactions, we're using energy to make larger molecules from smaller molecules. And again, it takes energy to form these molecules, to hold all of these atoms together. Catabolic reactions are the opposite. So we start with a larger, more complex molecule, and we break it apart. We break all these bonds, and we liberate the energy that was holding the atoms together, and we're left with smaller molecules. Metabolic pathways are the sequences of all the chemical reactions in a cell. So these chemical reactions are catalyzed by those enzymes. So this means your metabolic pathways are determined by enzymes. So this is an example of one metabolic pathway using enzymes. So these pathways are kind of like a chain set of reactions. So every enzyme is one link in the chain. So we have to go in this stepwise fashion to get our end product. So we can't just go from point A to point D. We have to go to all of these intermediate steps along the way. So enzyme 1 produces a product which activates enzyme 2, which produces a product to activate enzyme 3. So we get our final product. Metabolic pathways are genetically determined because enzymes are proteins, and all proteins are encoded by genes in the DNA. The basic dogma of biology is that DNA is transcribed or copied into RNA and then translated into proteins. So this is how we go from the recipe in a cookbook in our DNA to an actual three-dimensional functioning protein or a cake that we can eat. Collision theory states that chemical reactions occur when atoms, ions, and molecules collide. So in order to form these molecules and have these chemical interactions, these atoms and molecules have to have a physical contact. So they have to bump into each other. And they have to bump into each other at an effective rate. So if it's not strong enough, right, they won't have enough force to have that reaction. And they also have to collide at the proper angles. So this activation energy is the collision energy that's required for the chemical reaction to occur. Reaction rate is the frequency of collisions. And do they contain enough energy to bring about that reaction? So we said enzymes were biological catalysts, so they help speed up these chemical reactions. So Enzymes can increase our reaction rate. Some other things that can increase chemical reaction rate include temperature, pressure, or concentration. So in this example, we have a few molecules, right? So they're pretty spaced out apart. There's not as many, so there's not as much opportunity for them to interact and collide. So the rate of reaction is low. However, if we increase the concentration, so we have more of these molecules, there'll be greater chance of collision, and the reaction rate is greater. So we said enzymes are biological catalysts. So a catalyst is just something that speeds up a chemical reaction. Enzymes are biological catalysts, right? Meaning that they're genetically encoded and synthesized by living cells. Enzymes are very specific, so they act on a specific substrate. So it's kind of a lock and key mechanism where only that specific shape of molecule or substrate will fit and activate our enzyme. And these enzymes speed up chemical reactions by lowering the amount of activation energy required. So this allows them to speed up reactions without increasing temperatures. Increase in temperature can speed up a chemical reaction, but if you increase the temperature too much, you can start to damage the cell and its proteins and structure. So this is showing two reactions. So without the enzyme, right, we have a much steeper hill to overcome before we can get to our end product. But with the enzyme, we lower the amount of energy required. So we have a less steep hill. So we don't have to input as much energy to get our ball rolling in our end products. We said enzymes are specific for their particular substrate. So only a specific shaped molecule will fit into the active binding site of our enzymes. When the substrate contacts the enzyme's active site, it forms an enzyme-substrate complex. Right? So now we have a substrate bonded to our enzyme. So from here, the substrate will be transformed or rearranged into the product. So our chemical reaction will now occur. So once it's completed, we have our end products. We release the products from the enzyme. So at the end of this process, the enzyme is unchanged. Right? So it is free to react with other substrates. So another substrate can come along and bind, and it can continually produce these products. Names of enzymes usually end in ACE, A-S-E. So if you ever see ACE at the end of a word, right, that's a hint that we're talking about an enzyme. And they're usually named and grouped based on the type of reaction they catalyze. So an oxidoreductase enzyme is going to be involved in oxidation-reduction reactions. Transferase enzyme is involved in transferring functional groups between molecules. Hydrolase is a hydrolysis enzyme. Ligase is an enzyme that's involved in joining two molecules together. So it kind of ties molecules together. So it forms a ligature, right, or a ligand. So ligase, it forms a ligature or a tie for two molecules. Looking at enzyme components, an apoenzyme is the protein portion. So by itself, it's inactive. Right? So it needs a cofactor, which is a non-protein portion, to activate that enzyme. So once we have our cofactor, now our active site is the right shape for our substrate. So see, before we had that coenzyme or cofactor, right, our substrate's not going to fit in this spot. So coenzymes, cofactors could be things like vitamins and minerals. Right? So if it's an organic cofactor, it's referred to as a coenzyme. So the entire whole active enzyme is called the holoenzyme. So it's your apoenzyme plus the cofactor. So now our active site is the correct shape, and this is an active enzyme. There are a few factors that can influence the rate of enzyme activity. So things like temperature, pH, concentration, or inhibitors. High temperature and extreme pH can influence enzyme activity because we can denature those proteins. So remember, enzymes are proteins. And think back to anatomy, kind of the underlying theme in anatomy and physiology was that structure reflects function. right? So even down to the molecular level. So the structure, three-dimensional structure of that enzyme protein is going to dictate its function, how it catalyzes those chemical reactions. So if we denature or unwind and uncurl these protein fibers, it can no longer function in that metabolic pathway. So generally, once proteins have been denatured, right, it's an irreversible process. Right? So when you fry an egg, the egg white is basically pure protein called albumin. So the increasing temperature from cooking the egg denatures those proteins, and that's when it gets that solid white appearance. So once you fry that egg, you can't unfry it. So those proteins are permanently denatured. Temperature can increase enzyme activity up to a certain point. So the higher the temperature, the faster particles and molecules can move. So the faster they can move, the more likely they are to collide with enough force to have a reaction. Right? But again, once we reach that tipping point, so now at this temperature, the proteins will start to denature and enzyme activity will fall dramatically. High pH can also denature proteins. So just like most things, there is a sweet spot on the pH scale of where enzymes function best. So enzymes are most active around 5 pH. So once we get outside of that acceptable range, then our enzyme activity will drop off. So either it's too acidic or it's too basic. If the concentration of the substrate is high enough, the enzyme can catalyze at its maximum rate. Right? So at this point, all of the available enzymes are saturated or filled with substrate. So we can't increase our enzyme activity anymore unless we make more enzymes. Inhibitors are things that can inhibit the function of an enzyme. Competitive inhibitors are going to compete with the normal substrate and fill those active binding sites. So normal binding of a substrate, right? So it fits in there just like a perfect little puzzle piece. With a competitive inhibitor, it's competing for that active site. So with the competitive inhibitor in place, so if it gets there first, now our normal substrate can no longer fit in that active site. So some antibiotics and antimicrobials work as competitive inhibitors. So if we can turn off the function of a bacteria's enzyme, so it can no longer carry out its biochemical and metabolic reactions. So typically, competitive inhibitors are molecularly similar to the normal substrate. So see here, sulfonamide would be the inhibitor of our normal PABA molecule. So they're pretty similar, just a few minor differences, right? But it's similar enough that it can fit partially in that active site and block our substrates. Non-competitive inhibitors also inhibit an enzyme from functioning, but instead of binding to the active site, they interact with another part of the enzyme called the allosteric site. So it's kind of a backdoor binding site on the enzyme. So this is called allosteric or non-competitive inhibition. Right? So this inhibitor is not competing with our normal substrate. Non-competitive inhibitors work by indirectly changing the shape of the enzyme. So again, back to structure reflects function. If we change the shape of the enzyme, it's virtually non-functional now. So these types of inhibitors can be either reversible or irreversible. So again, competitive inhibition, we're interfering with the active site, so the substrate cannot bind. Non-competitive inhibition, we're binding to the allosteric site and changing the shape of the enzyme so the substrate can't bind. Feedback inhibition is when the final end product of a reaction acts as an allosteric non-competitive inhibitor to inhibit the enzyme from earlier in the pathway. So kind of a negative feedback loop. So if you think back to anatomy and physiology, negative feedback loops are how we maintain homeostasis. So in this example, we have our metabolic pathway, right? So it's a series of enzymes kind of in a chain. So our first enzyme, right, binds to the substrate, produces a product, right, which is a substrate for another enzyme, produces a product, right? So we get our final end goal product that we wanted. So now that we have our product, our enzymes, our metabolic pathway has done its job, right? So now we just go back and turn off that enzyme. So it's kind of like a thermostat. So once we reach the levels we need, right, once we reach the right temperature, right, we feed back into that pathway to turn off that initial source. Oxidation-reduction reactions are common coupled or paired reactions. Oxidation involves the removal of electrons. Right? So this compound A has some electrons here. It's been oxidized, and now it has no more electrons. Reduction is the gain of the electrons. Right? So the electrons that were oxidized from A have now been reduced to B. So compound A has been oxidized, compound B has gained electrons and been reduced. So you can think of it in terms of the charge. Remember, electrons have negative charges. So the compound that receives the electrons has been reduced because its electrical charge has been reduced. So it's more negative now than it was before. These reactions are sometimes referred to as redox reactions. So it's where we have that coupled or that paired reaction. So they occur kind of simultaneously, right, swapping those electrons. ATP is generated by phosphorylation of ADP. So phosphorylation is just a big word to say adding a phosphate group. Right? So ATP stands for adenosine triphosphate. Okay, so 1, 2, 3 triphosphate groups. ADP is diphosphate, right? So it only has 2. So ATP is generated by adding a phosphate group to ADP. Right? And remember we said those anabolic reactions, so when you add complexity to a molecule, you add atoms or molecules together, it's going to require energy to form those new chemical bonds. So ADP, diphosphate, plus a little bit of energy and a free phosphate group, and we have ATP, which is the major energy molecule of the cell. So now when a cell needs to use energy for something like active transport, the energy from here is now stored in this chemical bond. So when we break this bond in a catabolic reaction, we free up that energy to power the cell processes. Substrate level phosphorylation is just an enzyme produced ATP. So ADP and a substrate acts on the enzyme, right, binds to the active site. The enzyme catalyzes the chemical reaction to transfer our phosphate to make ATP. Oxidative phosphorylation is when electrons are transferred down an electron transport chain. It releases energy to generate ATP. So we'll have to go back in just a little bit from the beginning of this process of ATP and energy production. Kind of at the end, we cash in all of the electrons and energy that we've harvested from breaking down more complex molecules via catabolic reactions. And these energized electrons are passed from protein to protein, kind of hot potato style, along the membrane. So every time they're passed from one protein to another along this electron transport chain, it releases little bits of energy. Those little bits of energy are used to actively transport hydrogen across the membrane. So remember, active transport involves going from a low to high concentration. So we're going against the normal flow or gradient. So once we establish this hydrogen gradient outside the membrane, chemiosmosis is the process where ATP is generated using the energy derived from this newly formed gradient. So we pumped all of these hydrogens across the membrane. We're using active transport, so now they're going to passively diffuse back across the membrane through an enzyme called ATP synthase. So remember we said enzymes are typically named for what they do. So ATP synthase is an enzyme, ACE, that synthesizes ATP. So as these hydrogens flow through this enzyme, it's going to kind of spin like a turbine. So kind of like how a hydroelectric dam works. Right? So this would be the water behind the dam causing this to spin, and it's going to provide the energy we need to phosphorylate our ADP and make ATP. Photophosphorylation occurs with photosynthesis. So it involves light-trapping pigments like chlorophylls, right, within the cells and the chloroplasts. This light energy is going to be converted into a chemical energy in the form of ATP. So similar to oxidative phosphorylation, we have an electron transport chain, and that's going to pump the hydrogens right across to have the chemiosmosis to produce ATP. There are two main metabolic pathways for energy production that we'll look at, but both are a series of enzymatically catalyzed chemical reactions. They're going to extract energy from organic compounds, so things like sugar, and store it in chemical energy of ATP. So in this chapter, we'll look at cellular respiration and fermentation. The breakdown of carbohydrates, things like glucose and sugars, to release energy typically occurs in 3 stages for cellular respiration. So glycolysis is the splitting of sugar. The Krebs cycle is where we're going to harvest most of our energy carriers in this process. And the electron transport chain is where we have that oxidative phosphorylation. So all of these energized electrons, right, we're now going to cash in at the very end, right, and they're passed from protein to protein, pumping hydrogen, right, and then the hydrogen flows through, generates ATP. So cellular respiration is also referred to as aerobic respiration, meaning with oxygen. So in cellular aerobic respiration, oxygen is the final electron acceptor. Right? So this just means once our energized electrons have reached the end of the chain, they've pretty much been zapped of all of their energy. They've been depleted. So we need to remove them from the chain to keep the energy flowing. So oxygen will bind to those depleted electrons to remove them from the chain and form water. Glycolysis is the first step in cellular aerobic respiration. So it just means splitting sugar. So lysis means to split and glyco means sugar. So glycolysis, we start with a 6-carbon glucose molecule, and through a series of reactions in our metabolic pathway, we end up with 2 3-carbon pyruvate molecules. There's 2 phases of glycolysis. In phase 1, we have to invest a little bit of energy just to get the ball rolling. But if everything goes well, then we should have a net profit of energy at the end. So at the beginning, we have to input 2 ATP molecules to begin the process. In phase 1, glucose is split into 2 G3P molecules. And also notice we're not changing the number of carbon atoms. We're just kind of rearranging some of these molecules. So in phase 2 is our energy payoff or energy conserving stage. So those 2 G3P molecules we just got, right, will now be oxidized into our pyruvate or pyruvic acid molecules. So during this process, we have produced a total of 4 ATP, which gives us a net gain of 2 ATP. So we have to pay back the 2 ATP we used to initiate glycolysis, right? But then we get a 2 ATP profit or net gain. We'll also capture 2 NADH molecules, which are our energy carriers. So every time these molecules, these chemical bonds are broken or rearranged, little bits of energy is given off. So instead of just losing that energy to the environment or as heat, we capture the energy in electrons. So this is what we're going to hang on to until we get to our electron transport chain at the very end. So just another angle of looking at glycolysis. So your energy investment stage, we invest 2 ATP, right, to form 2 G3P. So we're going to split the glucose in half, right, splitting sugar, glycolysis. So energy harvesting, our 2 G3P become 2 pyruvate. Right? And in the process, we're going to get some more ATP from this and a couple of energy carriers. In cellular respiration, the oxidation or removal of electrons from molecules is going to liberate or free up those electrons so they can then later operate in that electron transport chain. The final electron acceptor to remove those energy-depleted electrons from the chain come from outside the cell and is inorganic. In aerobic respiration, we said oxygen was the final electron acceptor. So oxygen will bind to those depleted electrons and just form a harmless waste product of water. Anaerobic respiration occurs without oxygen, so it has to use something other than oxygen for its final electron acceptor. The end result of oxidative phosphorylation is the production of ATP energy. So in previous chapter, we talked about the organelles and the parts of a cell. And we said the mitochondria was the powerhouse of the cell because it produces ATP. Well, it just means that this process of cellular respiration occurs inside the mitochondria. Krebs cycle is the second step of cellular respiration. So during the Krebs cycle, pyruvic acid is going to be oxidized into a 2-carbon molecule. Our third carbon that's cleaved off will just be given off as CO2. So this is why we exhale CO2 when we breathe in oxygen. So our remaining 2-carbon compound binds to coenzyme A and forms acetyl-CoA. So acetyl-CoA is what's going to enter into the Krebs cycle. So throughout different steps along the way of the Krebs cycle, we're going to harvest more of these energy carriers and get a little bit more ATP. So this is just showing more in-depth figure of the Krebs cycle. You don't need to know all of these steps. Just know our pyruvic acid that we got from glycolysis is going to be converted to acetyl-CoA. So pyruvic acid by itself is not able to enter the Krebs cycle. So we have to modify it a bit into acetyl-CoA. The Krebs cycle is sometimes also referred to as the citric acid cycle because citric acid is the first molecule that's made in this process. But as we continue to change the molecule and rearrange some of these atoms, right, we're collecting energy, energized electrons along the way. So again, remember all of these energy carriers we're going to hang on to and then cache them in the electron transport chain at the end. The electron transport chain occurs in the plasma membrane of prokaryotes or that inner mitochondrial membrane in eukaryotes. It's that series of carrier molecules or proteins that are oxidized and reduced as electrons pass down through this chain. So again, kind of a hot potato passing of the energized electrons from protein to protein. So every time we transfer those electrons, they give off little bits of energy. And that energy that's released is going to be used to produce ATP by chemiosmosis. So see, in glycolysis, we had 2 NADH. In the process of converting pyruvic acid to acetyl-CoA, right, our preparatory step, we got 2 more NADH. During the Krebs cycle, we got 6 NADH and 2 FADH. So total at the end of the process, when we're ready to cash in all of our chips, we have 10 NADH molecules and 2 FADH. So all these total should translate into a profit of 34 ATP. And then if we add in the 4 ATP we generated in the previous steps, we have a total of 38. Aerobic respiration is by far the most efficient means of ATP production. Chemiosmosis is when we're going to cash in all of our energy carriers, all of those NADH and FADH. So these energized electrons, as they pass from carrier molecule to carrier molecule, release energy. We're going to use this energy to pump hydrogens across the membrane via active transport. So energy from high energy electrons powers this active transport of hydrogen through the membrane. So now we've established a high concentration gradient of hydrogen outside compared to inside. So by law of natural passive diffusion, right, we want to go from high to low. So these hydrogens will now diffuse passively, meaning with no energy, through our ATP synthase enzyme. So the flow of hydrogen down its concentration gradient is going to power ATP synthesis, much like how water flowing can power a hydroelectric dam. Again, the final electron acceptor is oxygen. Without oxygen, our depleted electrons would be stuck in this chain. So here's our energy carriers, release energy to pump hydrogen, and then we pass the electron to the next protein. Releases more energy to pump more hydrogen. Same thing down the chain. So now at the end of the chain, this electron is depleted. So it binds to oxygen, forming water. And now it's exited the chain and the flow can continue. So now our new hydrogen gradient diffuses down ATP synthase, provides the energy to form that third phosphate bond of ATP. Anaerobic respiration occurs when there's no oxygen present. So this is also referred to as fermentation. The final electron acceptor in their electron transport chain is not oxygen. So while they still can produce some ATP, it's not as efficient as aerobic respiration. We don't get nearly as much ATP at the end of fermentation as we do aerobic respiration. Fermentation is similar to cellular respiration in that it's going to release energy from the oxidation of those organic molecules like glucose. Where it differs, though, is that it does not require oxygen and it does not use the Krebs cycle or the electron transport chain. The final electron acceptor in fermentation is typically an organic molecule, and we only get small amounts of ATP. So fermentation is essentially glycolysis just repeated. So remember we said with glycolysis, we do get a net gain of 2 ATP. So we have to invest 2 ATP, but then we get 2 at the end paid off. So for every round of fermentation, you basically only get 2 ATP, as opposed to with cellular respiration, aerobic respiration, we got like 38 ATP. So with fermentation, we split the glucose into pyruvate, right, just like we did before. But now, since there's no oxygen, we can't go down the normal aerobic pathway. So we go down fermentation pathway and produce ethanol, carbon dioxide, or another type of fermentation produces lactic acid. But cells can continue this process of fermentation as much as needed because we're essentially regenerating the components necessary to begin the reaction. So for every cycle of fermentation, we get 2 ATP. Lactic acid fermentation produces lactic acid. So our human cells are capable of lactic acid fermentation. So when you're exercising really hard and your muscles are sore and burning, so that's when your body switches into that anaerobic metabolism. So in a pinch, right, 2 ATP are better than 0. Right? But unlike natural fermenting organisms, we can only undergo fermentation for a short period of time because that lactic acid will build up in our cells and cause that paining, burning sensation. Homolactic fermentation produces only lactic acid. Heterolactic fermentation produces lactic acid and other compounds. But the process of glycolysis is essentially the same. So glucose is oxidized or broken down into pyruvic acid and then reduced by NADH. Alcohol fermentation produces ethanol and CO2. So we use fermentation for a lot of our own food purposes. So when you bake bread, right, what makes the bread rise, the yeast, is producing CO2 gases. Same thing with Swiss cheese. So the holes in Swiss cheese aren't cut in there. They're the remnants of CO2 gas bubbles during the fermentation process. Also with beer. So why beer is carbonated? Because during fermentation, all of that CO2 is accumulated. So this figure is showing the difference between a dry wine or a sweet wine. So what makes a wine dry or sweet? So in a dry wine, the yeast eats the sugar, but eventually they run out of sugar, right? And the yeast will start to starve. In sweet wine, the yeast eat the sugar, but there's more sugar than the yeast could ever eat. So as they continue to eat more and more sugar, they're producing more and more wastes. So eventually the yeast will be poisoned by their own waste, and there's only sugar remaining. So the sweet wine, there's still sugar left behind. The dry wine, the yeast have eaten up all the sugar. But either way, wine drinkers win. So you enjoy your delicious yeast poo and yeast corpses. You enjoy your next glass of wine. This table is just showing some industrial uses for different types of fermentation. So ethanol fermentation, right, we use to make beer, wine. And it's made by that Saccharomyces cerevisiae. So we mentioned cerveza means beer, right? So this is a sugar digesting yeast that makes beer. Lactic acid fermentations used for cheese, yogurt, bread, sauerkraut. So this is what gives just plain yogurt that kind of acidic, sour taste. And there are some other pharmaceutical, industrial uses, flavorings, fuels, and vitamins. So we can use biochemical tests to help identify bacteria by detecting specific enzymes. So again, enzymes are genetically coded. So not all bacteria will have the same genes or the same enzymes. A fermentation test tests for bacteria that can catabolize or break down carbohydrates or proteins and produce an acid. So if they're able to break down those nutrients and change the acid content of the solution, the pH indicator would change color. We can also test for fermentation by looking at a Durham tube for gas production. So if there are CO2 bubbles forming, an oxidase test identifies bacteria that have specific enzyme called oxidase, cytochrome C oxidase. So if the enzyme is present, it will be able to oxidize the reaction and give a positive result. If the oxidase enzyme is not present, the chemical reaction won't occur and there won't be a result. Photosynthesis is also composed of a coupled reaction. So we said anabolic and catabolic reactions are coupled. So basically the end product of one set of reactions can be used as the ingredients for a second set of reactions. So photosynthesis has two types of chemical reactions we'll look at. The light-dependent, or sometimes just called the light reactions, involve the conversion of light energy, electromagnetic radiation energy from sunlight, and we convert that into a chemical energy of ATP and NADPH. Light-independent reactions, sometimes referred to as dark reactions, even though they don't necessarily have to occur in the dark, they're just independent of a light being present. So light-independent reactions use the ATP and NADPH that we got from our first set of reactions, and they're used in the second set of reactions called the Calvin cycle. So our first set of reactions generates energy or converts energy from the sunlight. So we have usable chemical energy to power the production of glucose. So with photosynthesis, the organism takes carbon dioxide just out of the atmosphere and rearranges the molecules, breaks and forms new chemical bonds to give us a final product of glucose, which is a much more complex molecule. So if you really look at it, cellular respiration and photosynthesis are the same chemical reactions, just in reverse. So we have sugar, glucose, as an ingredient for cellular respiration. So we're breaking down glucose to make energy. So the reactants, glucose and oxygen, are the products in photosynthesis. So at the end of photosynthesis, we get glucose and oxygen. The products of respiration are CO2 and water, as well as ATP energy. So we exhale CO2 and we just form metabolic water. Well, photosynthesis ingredients, to undergo photosynthesis, they need CO2 from the atmosphere and they need water. In cellular respiration, we're generating ATP energy. In photosynthesis, we're inputting energy from sunlight instead of ATP. The light reactions in photosynthesis contain an electron transport chain very similar to what we saw with cellular respiration. So light energy energizes and stimulates the electrons from these photosystems. So this contains the chlorophyll green pigments that absorbs that energy. Right? So it energizes the electrons. Electrons are then transported to the electron transport chain. So just like we saw with oxidative phosphorylation, every time the electron is passed from protein to protein, kind of hot potato, one to the next, it releases little bits of energy. But instead of pumping the hydrogen out of the membrane, we're going to pump the hydrogens inside the membrane, so in the thylakoid space. But the same concept as before, we now have a high concentration gradient of hydrogen on one side of the membrane. So then via natural passive diffusion, the hydrogens will flow through that ATP synthase enzyme to generate ATP. So the ATP in our energy carrier NADPH that we gained from our first set of light reactions are then going to be used to power our second set of reactions in the Calvin cycle. So using the ATP energy in NADPH from our light-dependent reactions here, we're going to fix carbon dioxide from the atmosphere. So we take CO2 out of the atmosphere and we rearrange those molecules and those chemical bonds. So the end result of the Calvin cycle is a more complex molecule. So remember, when you're building a more complex molecule, it requires energy. Right? So this would be your anabolic type metabolism. So we need that energy to form those new bonds. So now the glucose that we've made in photosynthesis can enter into the cellular respiration cycle, where it will be broken down to produce ATP. One way we can classify organisms is based on a nutritional classification, so based on their primary energy source and their primary source of carbon. So because all living things are made of carbon, right, we're carbon-based life forms. If you ever take an organic chemistry class, it's nothing but carbon-based chemistry. So all living things have to have some source of carbon to form the backbone of their biological molecules. So some organisms may use a light source for their energy. So these are phototrophs. If an organism has a chemical energy source, it's a chemotroph. As far as carbon source, if the carbon source is from CO2 in the atmosphere, they're considered autotrophs because they're able to undergo photosynthesis. So if you can't undergo photosynthesis, you have to get your carbon from organic compounds. So meaning you have to eat other living things to harvest their carbon. So these are called heterotrophs. So hetero means other. So you have to eat other organisms to get your carbon. Whereas auto meaning self. Troph means feeding, so self-feeding or other feeding. Phototrophs use light energy to drive ATP production. Photoautotrophs bear carbon from CO2, so they undergo photosynthesis. So they use the CO2, they fix it in the Calvin cycle to make glucose sugar. Oxygenic photoautotrophs produce oxygen. So standard green plants, algae, and cyanobacteria, or photosynthetic bacteria. And oxygenic photoautotrophs do not produce oxygen. So these are more rare. Photoheterotrophs use organic compounds for their source of carbon. So they use light energy, but they still have to eat other organisms. So an example of a photoheterotroph would be a purple non-sulfur bacteria. Chemoautotrophs obtain energy from inorganic chemicals and use CO2 for their carbon source. Because they use CO2 for their carbon source, they still undergo that Calvin cycle to fix the CO2 into glucose. So these type of organisms can live off of things like nitrogen and are more common in your soil bacteria. So the difference between your chemoautotroph, like a soil bacteria, versus a photoautotroph, right, would be your standard plants. Chemoheterotrophs obtain energy and carbon from organic chemicals. So these are the most medically and economically important organisms. So this includes all animals, most fungi, protozoa, and bacteria. So all of the bacteria that we'll deal with and talk about in this class and lab would be chemoheterotrophs.